Bacterial cell walls are composed of one or two bilayers and a peptidoglycan shell. So gram-positive bacteria are ones that have one membrane, and then they also have a thick peptidoglycan outer layer. Gram-negative, on the other hand, has two membranes, and there's a thin layer of peptidoglycan in between. But what's important here is the structure of peptidoglycan. So while we could spend time talking about the differences between gram-positive and gram-negative bacteria, that's something that's better saved for biology courses where we can talk about gram staining and how all of that works. We're going to ignore all of that and focus just on the structure of peptidoglycan. So peptidoglycan does still have carbohydrates. We'll talk more about those in a minute. For now, I want to actually focus on this tetrapeptide. So if you'll notice, there's 4 different peptides, including isoglutamate, which doesn't have the normal linkages you usually see in glutamic acid. So it has kind of an unusual gamma carboxyl linkage here. So and there can be some differences in this tetrapeptide. So while there is a lot of homology between similar ones across different species of bacteria, there can also be differences. Focusing more on the carbohydrate is made up of NAG and NAM, NAG being N-acetylglucosamine, NAM being N-acetylmuramic acid. So if you'll notice, there's part that's not highlighted by the tetrapeptide that's hanging out above that L-alanine. That actually belongs to NAM. So NAM actually has a carboxylic acid group as part of that monosaccharide unit, and that's what is then forming the covalent bond to the tetrapeptide. To take a closer look, when we're looking at something that's gram-positive, to go in between all of the tetrapeptides, you actually have a pentaglycine crosslink. So yes, that is 5 residues of glycine stretching in between L-lysine to our D-alanine. And yes, this is also a good time to point out the fact that this is actually using both L and D versions of the amino acids. So here in this representation, we've lost some of the details of the individual elements and functional groups, but here you can again see NAG and NAM. NAG now represented by these reddish pink dots, NAM represented by blue, the tetrapeptide also represented by individual circles, and the pentaglycine crosslink also represented. So this represents a single layer of peptidoglycan. When we're looking at this, realize that the peptidoglycan layer essentially creates one giant molecule that is kind of bag-shaped that the bacteria then fits into. If we're looking at gram-negative peptidoglycan, the main difference here is there's a direct cross-link between that L-lysine and the D-alanine of different tetrapeptides. So there isn't that pentaglycine link in between them. They're directly cross-linked together. So it's a direct amide bond. When we are looking at these cell walls then, for gram-positive bacteria, they're going to have an inner lipid membrane layer, and then on the outside is going to be that peptidoglycan. So we sometimes can refer to that as a polysaccharide coat because we do essentially have not only these repeating units of those carbohydrates of NAG and NAM, but there's also multiple layers of peptidoglycan. So what you see here are multiple layers. 3 of them are represented here of peptidoglycan. For gram-positive bacteria, you can have up to 20 layers of peptidoglycan. So this can be a fairly thick sugar coat. And it is this peptidoglycan layer that then will react with gram stain, which by the way is purple. For gram-negative bacteria, we usually don't have nearly as many layers of peptidoglycan, and it's in between then 2 bilayers. So again, our peptidoglycan is represented by that orange bit that's in between, and our 2 bilayers are hanging out with those teal and purple colors. And then on the outside of our gram-negative bacteria, they also then have lipopolysaccharides. So those are actually going to be additional sugar chains that are then connected to a lipid that's in part of the bilayer. So you still have even more carbohydrates dealing in gram-negative bacteria. Talking more about that lipopolysaccharide. So again, we're going to have a lipid group that's going to be anchored to the outer outer membrane, and then that's going to be joined to a polysaccharide. It's going to have long chains with repeating subunits. can often refer to those as antigens because this is often going to deal in some of the recognition of those bacteria. So when we talk about more than just peptidoglycan, we can also have glycoproteins, which these can be either N-linked or O-linked. So if we're going to have some sort of carbohydrate attached to our protein, there has to be some sort of amino acid that it's linking to, because it's not going to particularly just link to either the C-terminal end or the N-terminal end. It's going to be linking to a side chain of an amino acid, because whenever we say glycoprotein, it's going to have usually many sugars attached to the outside. So N-linked glycoproteins are usually attached via the amide nitrogens of asparagine residues. O-linked saccharides are going to be attached with hydroxyl groups of either serine, threonine, or hydroxylysine. So what you see below is this is an O-linked saccharide, where what you have is something that is then attached to the functional group of the serine. Here then are showing particular ones of xylysylthreonine and mannosylserine. So again, these are also showing O-linked. So, but the one on the left is for threonine, the one on the right is for serine. This then is finally showing an N-linked, which yes, just is to asparagine. And so this actually has a more complicated oligosaccharide, but then again is N-linked, and N-linked because it's linked to asparagine. Glycoproteins, N-linked glycoproteins come in 3 main flavors. So since they actually have fewer options because they're only going to be linked to asparagine, you're going to have 3 main versions. One that is high in mannose, which mannose is just a particular unit that your polysaccharide can be made out of. Complex, and a hybrid, where you basically find combines the structures that are found in high mannose and complex. So when you look at the high mannose, what you do see is you see a lot of the sugar mannose over and over and over again. It's not only made up of mannose, but has a lot of mannose in it, especially on those outer layers. Complex then has a variety of different things. So complex just means you have a lot of different things, not just mannose. And hybrid then what you see is kind of a mix between the two, where there is a fair amount of mannose on one branch, but on the other branch not nearly as many. As you might imagine, this lecture on carbohydrates has just kind of been trying to give you a feel for some of the different places that you see carbohydrates. It's a bit difficult just because we could have gone into a lot more detail on the carbohydrates and how they're used in some of the interstitial fluids in the body. We could have spent a lot more time about how peptidoglycan works. We could have spent more time looking at some of the different glycoproteins and what some of those carbohydrates do on those proteins. So realize that this lecture, for which often happens in biochemistry, is a bit of an introduction to how they're used. So kind of keep your eyes peeled, because sugars are actually really interesting the more you're on the lookout for where they're having interesting functions.